and welcome to our integrative activity number 3. In this video, we will talk about DNA, RNA, and coded message we can form with this. First, we have to know how can we decode the message. To decode the message, we have to translate from DNA to RNA. Then, we took a protein and with this, a letter. With these letters, we write the message. The next one is the function of the codons. The codons are a mechanism of the body to isolate a part of the RNA. With that part, we can synthesize some proteins, and with that, we can write the message we talk about. Finally, which letters are not present in this message? The letters that are not present in this message are B, J, O, U, X, and C. These letters are not present in the message because they don't have a protein associated, so we cannot write with them. It's very simple, but also kind of interesting, we know. So let's go to the next part.
So the first question of part 5 says, how do you know which databases must use when you transcribe the DNA sequence to messenger RNA codons? Well, the answer is very easy. In transcription, the messenger RNA should be formed by a complementary base pairing of the template DNA strand. So the messenger RNA strand would be formed by matching the A in DNA with U in a messenger RNA, T with A, C with G, and G with C.
T with A, G with C, A, G, C, U because it's messenger RNA, E, C, U, A, C, and U.
The last question says, what step could add after transcribing the DNA to develop a more complete model of protein synthesis? Well, the answer is translation, which is a process that has a function to complete the protein synthesis model. So, the number 3 says, suppose some codons to different amino acids are mapped. What would be the effect of your translation of codon messages? What would be the effect on protein production? The answer is, if some codons were mapped to different amino acids, the translation of codon messages would not be accurate, because the protein that would be formed during protein synthesis would not have the same composition and nucleic acids, since the codons don't have a single amino acid. Number 4. During the real production of proteins in a cell, what will happen to an RNA chain before leaving the nucleus? The answer is, during the actual production of proteins in a cell, eukaryotic RNA are covalently altered before it leaves the nucleus. The transferred RNA are also altered before it can leave the nucleus. So it has to be synthesized by RNA polymerase 3 enzyme to produce mature transfer RNA.
Okay, so we have part A. In part A, we're going to have to write a messenger RNA complementary to the given DNA that we have. This is the given strand. So first we have to know that for each nucleotide we're going to have to write its opposite nucleotide. For example, for a T, we're going to have an A. But for an A, instead of a T, we're going to have a U. Then for a C, we'll have a G. For an A, we're going to have a U. Then for a T, we'll have an A. Then another U. Then for an A, we're going to have another U. Then a G. Then for a T, we have an A. And so on. Then we're going to have our second question. It's very clear. So we're going to have to use figure number 1, in which you have an amino acid and a letter. So we just have to write everything and write a word, which is make.
What is this? Angela, you know what is this?
No. A letter?
Oh my god! Look at this.
Maybe it's a secret message.
Wow.
We have to find like the amino acid by the DNA, maybe.
Gee, so we can know what it says.
Okay, let's do it.
Oh my god.
Okay, so maybe you need first to pass the DNA to RNA, no?
Oh yes, I think yes. So So this goes like G, and this one also G, and this is U, right?
Yes, because in RNA T, it's U.
Okay. So
Each codon represents a specific amino acid, and each amino acid is represented by one letter. So, in this circle, you read from the center to each side of the circle. For example, GGU is for glycine, and glycine is represented by G. And GAC is glutamic acid and represented by A. So we write the letter.
okay.
Then we have and UUG is for stop. So we need to stop there. And we have to write another word. Wow, and now we know the message of your letter.
Wow, this is amazing. I like this.
Guess what? We are going to practice. We have some translation and traduction exercises. Here we are going to translate the DNA code to RNA, and then the RNA to polypeptides, and then we are going to use this chart to convert the polypeptides into letters. Let's go and start. Now using this chart we are going to obtain the code. Meet me at 5. Oh my god.